Today, we're diving into a really cool puzzle. How do scientists take the complete DNA of a bacterium, which gets shattered into millions of little pieces, and then put it all back together into a perfect, complete genetic blueprint? Let's get into it.
So I want you to imagine this is your job. This is honestly the best way to think about genome assembly. You see, DNA sequencing machines don't just read a genome from start to finish in one go. Nope. They basically shatter it, getting us millions of tiny, jumbled up, and slightly error-prone pieces that we call reads. And our job is to figure out how to put that shredded, typo-riddled book back together again.
Now, the official term for this puzzle solving is de novo assembly. De novo is just a fancy way of saying from scratch. We are literally building the entire picture from the ground up, using only the little bits of overlapping information on each piece. And yeah, it's like solving a massive jigsaw puzzle without even having the picture on the box to guide you. It's a huge computational challenge.
So, what's the end game here? What are we actually aiming for? Well, it's what scientists call a perfect assembly, and this isn't about getting it good enough. According to our source material, this means two things: 100% accuracy, 0 errors in the sequence, and total completeness. That means every single piece of DNA, from the main chromosome down to any little extra bits called plasmids, is assembled into one single, continuous, unbroken loop.
All right, so to solve this puzzle, the first thing we have to do is gather all the pieces. And it turns out the quality of those pieces is absolutely everything. It all starts with getting the DNA out of the cell. If you start with cruddy, broken DNA, you're pretty much doomed from the start. The goal is to get what's called high molecular weight DNA. Just think of long, beautiful, unbroken threads. Any damage or tangles at this early stage just makes that final puzzle way, way harder to solve.
Okay, so once we have that pristine DNA, we need the right tools to actually read it. This table kind of lays out the main players. You can see we've got a few different technologies. On one hand, you have Illumina, which gives us these really short but incredibly accurate reads. Think of it like a proofreader with a magnifying glass, checking every single letter. Then you have technologies like PacBio and Nanopore, which give us much, much longer reads. They're more like the architects who see the overall blueprint, which is crucial for getting the big picture right.
So let's just break that down, because it's a really important trade-off. The short reads, they are super accurate. They can tell you exactly what letters are in one little sentence. But they get totally lost trying to connect different pages of the book. The long reads, on the other hand, they can see how entire chapters fit together. They can easily jump across those tricky, repetitive parts of the genome that confuse the short reads. They build the main scaffold, even if they misread a few words here and there.
So what's the answer? Do you have to pick one? Nope. The modern gold standard is what's called a hybrid assembly. You don't choose, you use both. You get the best of both worlds. You use the long reads to build the main structure, and then you use the super accurate short reads to come in and fix all the tiny spelling mistakes.
Okay, so we've got our high-quality pieces, both long and short reads. Now for the main event, actually putting the blueprint together.
And this lays out the workflow perfectly. It's what's known as a long read first approach. First, you build a draft using only the long reads. This gets you the correct overall structure. Some really clever methods, like using a tool called Tricycler, will actually build a few different versions of the assembly and then merge them into a consensus to avoid any big structural mistakes. After that, you start polishing, first with the long reads, and then, the crucial final step, using those highly accurate short reads to fix all the tiny little typos.
And this visual shows you exactly why that long read scaffold is such a game changer. On the left, you see what an incomplete assembly looks like. It's a tangled, fragmented mess. That's what happens when the software gets confused by repetitive DNA. On the right, that's the goal. A single, clean circle for the chromosome and a few smaller circles for the plasmids. Without long reads to span those tricky parts, you're stuck with the mess on the left.
So that initial assembly gets us really close, but remember, we're not aiming for close. We're aiming for perfect. And to get there, we absolutely have to talk about polishing. This is where we go from good to great.
So you might be wondering, why is polishing such a big deal? Well, this quote from the source material really hits the nail on the head. The small errors that are pretty common in long reads, usually just a single letter being added or deleted, can cause what's called a frame shift. This completely messes up how the genetic code is read by our software. It's like taking a sentence and deleting one letter near the beginning. All the words that come after it just become gibberish.
And that is where our ultra-accurate short reads make their triumphant return. This step, short read polishing, is basically using those Illumina reads to meticulously go through the long read assembly and fix all those tiny insertion and deletion errors. We call them indels. It's the final proofread that makes sure every single genetic sentence makes perfect sense.
All right, so the assembly is built, it's polished, it looks amazing. But we're scientists, right? We can't just trust our gut. We have to verify everything. We have to check our work. And this is the million-dollar question, isn't it? How do we grade our own homework? How do we measure just how close to perfect we've actually managed to get? Well, thankfully, there's a whole toolkit of quality metrics we can use to do exactly that. So let's quickly run through these. First, you have metrics like N50 and L50, which basically measure how fragmented your assembly is. Is your final puzzle one giant piece, or is it 100 little pieces? The L50 is super simple. It's how many pieces it takes to cover half the genome. For a perfect chromosome, that number should be 1. Then you have tools like CheckM, which act like a parts inventory. It scans the genome to make sure all the essential, universal genes that are supposed to be there are actually present, and that there's no contamination.
And just to put a hard number on it, a high-quality assembly should have over 95% of these essential genes accounted for. This gives us a really solid benchmark for what success looks like.
Okay, so once we are absolutely confident that our assembly is top-notch, we finally get to the last, and maybe the most exciting step, annotation. This is where we finally get to read the book that we've so carefully pieced together. We use software to scan the genetic code, find all the genes, and predict what they actually do. This is what turns a raw sequence into real biological meaning. And that's the magic of it, right there. It's this amazing transformation from just a long, meaningless string of A's, T's, C's, and G's into the actual, functional blueprint for a living thing.
And that really brings us full circle, doesn't it? It summarizes the entire journey. We started with a complete chaotic mess of millions of tiny, broken DNA fragments, and through this incredible multi-step process of assembly, polishing, and quality control, we've reconstructed the complete, perfect circle of a bacterial genome.
You know, the ultimate goal here is to make this process so streamlined and reliable that it becomes fully automated. So I'll just leave you with this question to think about. When assembling a perfect genome is as simple as clicking a single button, what profound biological mysteries, from disease to evolution to antibiotic resistance, what are we finally going to be able to solve?